Hello everyone, I hope that you're all doing well. My name is Richard and welcome to our final presentation of the semester. So just a quick description, using the poster you have made over the past 4 weeks, your job will be to walk through the process for how nucleotides and ribose are made and show how these come together to make ATP used for DNA replication. So our goals, starting from glucose and CO2, were to walk through how nucleotides and ribose are made, and we need to describe how these come together to make ATP. So everything starts with the pentose phosphate pathway. So as you can see over here in the image on the right, it's an alternative branch off of glycolysis that produces the sugars that make up DNA and RNA. So in the PPP, no ATP is produced or used up. So in order to get to the PPP, you start with glucose, and you form glucose 6-phosphate via addition of a phosphate group, and the PPP can use any available molecules of G6P, whether they are produced by glycolysis or other methods. So there are two phases of the PPP, the oxidative phase and the non-oxidative phase. So the first two steps represent the oxidative phase. So in the first step, G6P is oxidized to form lactone. NADPH is produced as a byproduct. You can see here that NAD+ is reduced while G6P is oxidized. So another reaction catalyzed by a different enzyme uses H2O to form this molecule here, 6-phosphogluconoate. And then in step 2, a carbon is cleaved and carbon dioxide is released, and the product you're left with is ribulose 5-phosphate. So from this point onward, it is the non-oxidative phase of the PPP. That basically just means that these reactions are all reversible, and it allows for different molecules to enter the PPP in different areas of the non-oxidative phase, and they can be transformed up until the first molecule of the non-oxidative phase, which was the ribulose 5-phosphate that I mentioned earlier. So in the third step, the ribulose 5-phosphate is converted into ribose 5-phosphate, and notice that they have the same carbon count. This ribose 5-phosphate is very important because that's the sugar that's used to make DNA and RNA. And you can over here on the right, you can see the overall pathway, the overall PPP, and all the steps that go through. Oxidative phase, first two steps, non-oxidative phase, everything else. Ribose 5-phosphate made up here. So now that we've generated ribose 5-phosphate, we can convert it to form phosphoribosyl pyrophosphate, or PRPP. This is the overall equation here that does so. So ribose 5-phosphate reacts with ATP to form PRPP. And that's very important because PRPP is used in de novo nucleotide synthesis. And it can undergo two pathways, purine synthesis to form adenine and guanine, or pyrimidine synthesis, which forms cytosine, uracil, or thymine. Let's start with purine synthesis. So again, these form your A and G nucleotides. So I drew out a simplified pathway for purine synthesis. If you want more detail and information, you can refer to pages 4 and 5 of the poster that we made. So look at that for more detail. But basically, what you have here is PRPP reacts with the glutamine phosphoribosyl aminotransferase enzyme that forms your 5-phosphoribosyl 1-amine. Then it undergoes a lot of different reactions, and it will eventually form inosinate, or IMP. So this molecule IMP is very important because it allows you to form either adenylate or guanylate, which will eventually form ATP and GTP using the following steps here. So if you want to form GTP, for example, you take your IMP, you react with IMP dehydrogenase, you form xanthylate, you react with GMP synthetase, and you will form guanylate, which will eventually form into GTP. If you want to form ATP, which is what we want to do in this case, take your IMP, you'll react it with adenylosuccinate synthetase, you'll form your adenylosuccinate, react again with fumarate and adenylosuccinate synthetase, and you will form adenylate AMP, which will eventually form ATP. So now let's look at the other reaction, pyrimidine biosynthesis. So again, these form your C, U, and T nucleotides. So you start with bicarbonate and ATP. You react with carbamoyl phosphate synthetase. You'll form your carboxyphosphate. React again with CPS, you'll form carbamic acid, reacting with CPS, you'll form your carbamoyl phosphate. React with aspartate, you will form your carbamoyl aspartate. Then you'll form your dihydroorotate, and you'll eventually form orotate. And you'll react with the PRPP and pyrimidine phosphoribosyltransferase transferase, and you'll form this very important molecule here, orotidylate. So with orotidylate, you can react with orotidylate decarboxylase, and you'll form UMP. So this UMP, uracil monophosphate, you can form TTP, UTP, or CTP, which are all the pyrimidine nucleotides that you need. So if you want to form TTP, you just react with thiorodylase synthetase, and you'll form TTP. You react with UMP kinase to form UDP, then you react with nucleoside diphosphate kinase to form UTP, and then if you want to form CTP, you just react with CTP synthase, and that forms CTP. And again, please refer to pages 4 and 5 of the poster for more detail. So in conclusion, starting from glycolysis, we started with glucose, we formed glucose 6-phosphate, and then that allows us to go to the pentose phosphate pathway. One of the products of the PPP is ribose 5-phosphate, and this is a sugar used in the formation of DNA and RNA. Ribose 5-phosphate also leads to the formation of phosphoribosyl pyrophosphate, PRPP, which is important for de novo nucleotide synthesis. There are two pathways of nucleotide synthesis, purine, which forms A and G, and pyrimidine, which forms C, U, and T. And ATP is generated along this pathway. So it provides another method for ATP generation besides cellular respiration. And again, cellular respiration involves glycolysis, pyruvate oxidation, Krebs cycle, electron transport chain, and finally ATP synthase. So again, this is just a my poster that I created over the past 4 weeks. Hopefully it looks the same as everyone else's. So the pathway that I described mostly, which is the formation of ribonucleotides, that starts from uses pages 7, pages 1, pages 4, and pages 5. The other pathway to generate ATP, which was cellular respiration, uses pages 7, pages 12, pages 11, and pages 6. All right, thank you for the wonderful semester, and I hope you all have a great rest of the summer. Thank you very much.